Swayam Prabha. Digital India. Educated India.
Okay, so yes, so remember that a major issue in this effort of organization and everything is an expression system that will be completely digital instead of being okay, then you have to think, you have to something you don't think that selling daily is going to give each other or whatever, you're going to solve all your problems, right? That's automatic. And one possibility would be this. Okay. So what you want to have is a very, very strict control in expression and in interest. In this case, it's upper things, but we are already. So if you forget about these things here, what you have as an user, the user will activate a transcription factor. Transcription factor will activate a regulator. Then the regulator will activate interest. But what if we make a circuit, a rational circuit, in which at the same time that you produce R, then this R will produce a small RNA that will inhibit expression of R itself. What would be the consequences? Let me see what happens. Okay, so let's suppose that we organize this type of circuits. So we have the biosensor scheme as we had it before. So we have a factor, a box, collection, and this is inducible promoter model there. But then what we do is that connect that genetically to a device in which you do the following. The hopes that come from here go into a repressor. And the repressor is coupled to expression of the gene of interest. One part of it. And the coupling of this is important. I will tell you among ways. But then at the same time, this repressor represses expression of a small RNA here that inhibits in itself expression of this R here. The difference is that the repression of this repressor of this small RNA is transcriptional, whereas the repression of this small RNA into the regulator, this repressor, is just inhibiting translation. Okay. So there's another twist here, and it's the following. And it's that the expression of the gene of interest is coupled translationally to the expression of the repressor here. Why is that important? Because we have engineered the intergenic region between the repressor and the gene of interest in such a way that only if the repressor is expressed, then the gene of interest is translated. And the reason for that, in some cases, you can engineer a sequence in the DNA that gives rise to an RNA that falls in such a way that the ribosomal binding site will not be available to the ribosome. And only if the first gene is translated, it will unfold this region here, and then the ribosomal binding site of the second gene will be amenable to translation. This is one of the many tricks that are available to genetic engineers to modulate and to engineer to be some complex, say, control of some devices. So in this system, you have various components. You have transcription that is regulated, and then you have a repressor that is translationally coupled to a gene of interest, but then on top of that, you have a small RNA that will inhibit translational the expression of this R gene that in itself will repress expression of this small RNA. Biomedical is very complicated, yeah. It's true, but then we have the systems biologists, the mathematicians that say, well, look, I have this idea, what do you think? Will it work or it will not work? Then, well, about that, I don't want to do the details here, but we have calculator called Angel that is able to develop a mathematical model that show clearly that if you get the right set of parameters in this scheme, it will work. I mean, so it's like the percolator and the switch that we observed the other day. I mean, it's not enough to put the thing one after the other, but to get the right parameters. And we have the right parameters, chances are they. So if you control the strength of the promoter with the amount of small RNA, then there's a kind of set of parameters that will say that, well, you see that you are here, and then all of a sudden, what? It goes all the way up. So you have a digital response here. Okay. So the question is, well, finding this right set of parameters. So this is finally what we did. So this is the single promoter model and the digitalizer model. First of all, we look at whether it will work with proxy, like EFP protein. Of interest in this case will be a EFP protein. And well, this is what so you get this introduction, and then you got the expression of the EFP, and then you see that in fact, at least at a distance, it's very this is not induced. This is induced. There's no very detailed overlap. You cannot detect any overlap. You remember that we had this situation before, that in the first sight there was no overlap between the induced state and the non-induced state, and despite that we observed that there was some leakage in the basal promoter level. So how can we be sure that not even one molecule escapes the regulatory system? Well, an idea of how particular thing. Okay, so the only way, or one way to do it, is to engineer a system with the regulatory, with the molecule that may or may not escape is lethal. So there are some molecules called colysins that are able to kill a single cell just with one molecule. There are various colysins. The one that we pick is the colysins called E3. There are many of them, and there are RNases that leave 16 RNA regions, and then basically they kill the cells with just one molecule per cell. Okay, so the ultimate proof that you have a complete switch is in case where you ensure that the expression of a toxic protein is zero, because you know that if it is not zero, cells will not put that. Okay, so this is what we did, and we put some proxy, in this case not GLP, but called E3. And look what happens. In the absence of induction, cells are very, very happy here. In the moment you induce, cells die instantly. Well, they may recover after a while, quite. But cells die. Okay, you have a system in which you can really control expression of a molecule that works in such a way that only one molecule kills this. If you have the situation, that means that the system is very robust and it's operates very well. And then count variables, or you say quantify growth. You see that the moment, I mean, instantly after you add the user, cells die immediately. But at the beginning, they are all perfectly viable. You can even have this in a very difficult form. So what you have on top is E. coli that has been transformed, a construct that carries this colicin 3. Then action, you see that happen. We use the system, and cells die for this halogen. By the way, you don't have a clear halo. What you see when you have this situation is that you have many bacteria or many clones that cease to have escaped the trick that you have imposed on them. Can you figure out what are these, say, colonies that escape the circuit? Well, you have the possibilities. They are positive mutants. That because it is so complex, you have one part. I mean, you count the number of parts here. You have one part, 2, 3, 4, 5, 6, 7. You have 8 parts. You have so many parts that any mutation, any gradual change in one of them will kill entirely the system. So further, these mutations can be spontaneous or can be reduced by what is called insertion sequences. The insertion sequences are segments of DNA that are in bacterial chromosomes that are jumping from one place to the other all the time. Transposons, in fact, are segments of DNA that are flanked by insertion sequences, but sometimes insertion sequences have the ability to move by themselves. Okay? So if you take a strain that instead of being a normally called strain, is a strain that has been deleted of all insertion sequences, then you see that you don't have this reversion. That's another kind of trick that we use all the time. Secure that we construct, it will remain in time and will be there. Okay, so finally, once we rethink the special system and everything, then we make the final experiment, the transcendental experiment, and is to have in this construct the protease that cleaves the enzyme of interest, and we want to see whether the control is upstream. And fortunately, the answer is yes. So this is what happens when you have the system that I showed you before, where there's control, but the control is not digital. You have a lot of noise here. And this is what happens when the control is digital. See that you can go here to here with no intermediation. So obviously, this is one of the days that you kind of relax, because until this point you are not sure that the experiment will work. I don't know how many of you work in laboratories, but work in laboratory is a super intense experience because you never know whether the experiments will work or not. So the day that they work, it's a kind of explosion of happiness. If they don't work, you get very depressed. This is one day that we were all very happy because indeed we had this digital response to Okay, so can this digitalizer be implemented with other promoters and repressors? So we have one very good tool to digitalize in expression, not only this particular project, but for many other projects. Well, what we did, and again, I don't want details, is to use different combinations of different regulators and different repressors to see whether we would obtain this digitalization, and the answer is yes. So this particular circuit that was developed on purpose to solve a specific problem can now be used to be generalized to solve a large variety of problems in which the question at stake is to have a system that is completely repressed under non-inducible conditions and super highly expressed under induced conditions. Okay? So you can change the repressor, you can change the promoter. So that is the case where you really see that the same circuit can be implemented with different parts. And this is another important concept in synthetic biology. You can have a circuit and you can make an abstraction of the circuit, but then you can implement it, you can materialize it in many different forms, and in this particular case, you can change the regulators, you can change the repressors, and the system, the circuit will still work very well. Okay, but remember that all I told you before was about how to bring about compartmentalization or orthogonalization of a given reaction in a super complex regulatory network, a metabolic network. So what else we can think on doing to bring this about, different of what we have done before? Okay, again, synthetic biology, maybe you can ask this to GPT chat and see what we have this problem. What do you think? Maybe they will come up with an interesting problem, but an interesting answer. But the other thing that we figured out that we could use were camels. And you say, well, what are you talking about? The camels. I mean, what camels have to do with metabolic engineering and orthogonalization? Okay, here comes again the creativity of synthetic biologists. Okay? So they never put limitations to their way of thinking or to propose extraordinary, out-of-the-box solutions to problems and so on. So, by the same token, that you say, well, I can inactivate enzymes that are adjacent to the reactions of interest with this conditional proteolysis. Well, maybe I can do the same, but instead of cleaving the enzymes, I perhaps can use a different strategy. And the strategy is to use camel antibodies. Why camel antibodies? Well, camels produce a type of antibodies that are naturally single chain. So you have studied biology a bit, you know that human antibodies and animal antibodies have the long chain, the short chain, and so on. And the business part of the antibody is this thing here, this is like the tip of the Y here. And well, this is the standard antibody. But camels have been known for a long time to produce, apart of the standard antibodies, another type of antibody that is much simpler and has a model that is the so-called the VHH model, that is a very small model, and it by itself has the capacity to inhibit, just to interact with the antigen of interest here. Okay? So we have this molecule here that by itself has a tremendous ability to touch another protein or another antigen of your choice. Then, what happens about these camel antibodies is that they are evolved naturally to inhibit enzymes, to inhibit venoms. There's a theory that camels develop this ability to avoid the venom from snakes, for instance. Okay? I don't know whether it's true or not, but what is the idea? But in any case, for instance, what you have here is a structure through which an antibody against lysozyme inhibits lysozyme activity, because there is this part of the antibody that protrudes from the protein structure as a little finger, and the finger will go inside the active site of the enzyme. So it's a super good enzyme inhibitors. The structures are such that they are very good at inhibiting enzymes. Okay? So there's a problem here, and is that they produce, to have activity, they have to be assembled with a disulfide bridge. You get into this nitty-gritty of synthetic biology. So, and you want this, say, protein to be active intracellularly, and the intracellular medium is always very reductive. So how can we produce these antibodies inside the cell so they can inhibit the proteins that we want to inhibit? How do we deal with this disulfide bridge? Well, years ago, we discovered that if you make a protein fusion between the structure of a camel antibody and thyroredoxin, then the antibody can be expressed intracellularly with full activity. Okay, so that was really good, because then you have a tool. Again, you don't need to go into the details, but there are ways in which you can express intracellularly camel antibodies, or the business part of the camel antibody that is called a nanobody, because it's very small. A nanobody, you can express that intracellularly, and there's no problem. You don't need to form a disulfide bridge. The other problem is that you need camels, and where do you find camels? Well, maybe in India you have camels closer, but for us in Spain, that's not so easy. So we had to look for the camels to the Canary Islands. So the Canary Islands are some islands that is Spanish territory off the coast of Africa here. And then they have a bunch of camels. They are used for tourists, so it's not that we use them for anything else. So it's a big business having camel herds. So we started a collaboration, this is myself, a few years younger, and then we started a collaboration with some camel farms in the Canary Islands to get camels inoculated. Camels here that have been exposed to the antigen, and then what you do is that you reconstruct the sequence that you have amplified in a phage display system. Phage display is a methodology, again, developed time ago. Recently, one of its creators received the Nobel Prize for that. And what it is that now you can express and display the protein structure of choice in the tip of a phage, in the tip of a M13 phage. And in this way, you can recover the protein that interacts with a given antigen without having to isolate the protein itself from the camel. Okay, so instead of isolating the antibodies from the camel, you capture the sequence and then you express the sequence in a phage that enables you to take the sequence and the protein to the laboratory without having to bother the animal any longer. Well, this has some early steps and other details. I will not go into detail, but finally, after some say rounds of selection in petri dishes and in microbacterial plates, at the end you end up with a number of enzymes. A number of antibodies that have been isolated in response to inoculating the enzyme in a camp, right? Okay, so I'll give you an example of how you can leverage that, how can you really exploit that circumstance for using, for making this type of metabolic engineering where you want to inhibit one specific step in a pathway. So, and you can do that in E. coli, but you can do that in any other bacterium. And in this case, what we did it in this bacterium here called Pseudomonas stutida. In other talks, I will tell you a lot of things about Pseudomonas stutida, but at this point, it's only just one more gram-negative bacterium of interest that is amenable and is interested to make some metabolic engineering. Okay, so here the problem, you remember the problem before was about hyperproduction of this hydroxy acetone phosphate. In this case, the problem is different. In this case, the problem is isolation of this step here. So we have nicotinic acid that is transformed by this bacterium into this other compound, and then this other compound phosphate, not a mutant in the genetic sense, but a system that will enable to control, to knock out phenotypically this NIPX protein. So how can you do that? This is the pipeline. So this is the entire pathway. This is the step that you want to inhibit that is regulated or is brought about by the enzyme called NIPX. So you purify, or we purify the X, that is this enzyme. Then we generated, we inoculated a column. We took the lymphocytes. We PCR up the corresponding sequences. We brought this sequence to a phase display system. The phase display system was, the phase display pool was pan against a surface that was coated with a protein here, and then eventually we were able to get the right clones, and these right clones were recloned and intracellular expression vector for these sequences there. So it went to Pseudomonas stutida. So you have to work all these steps. These steps are fastidious from the point of view, but now are, it's a pipeline. So a pipeline is that you one thing and then the other, and the other. So, and these pipelines are available in laboratories with us, but everything starts with having a camel inoculated with the enzyme that eventually you want to stop there. Okay, so at the end we obtain a good antibody against the X that is called G6VH8. Well, you can name it. And then it needs to be fused to thioredoxin to secure that when it goes into Pseudomonas, then there's the expression is right. So that's okay. Now you obtain many antibodies, and none of them will be inhibitory, right? So some of them you have a protein, and the antibodies will touch different parts of the protein, and obviously not all of them will be inhibitory. So you have to focus on those that when you express them in vivo are inhibitory. And this one in particular, the one called G6, was the only one or the best one that after some tests produced an inhibitory activity on the X. Okay, then you express is the concept inside Pseudomonas stutida and see how it behaves. Well, first of all, you can monitor that the protein is expressed, you can protein gel, you can identify the bands, the proteomics, or just acid or whatever. And then what you observe that this protein, this antibody expressed in Pseudomonas stutida, antibody cells with nicotinic acid, then here a considerable decrease in the activity. And this is also manifested here. So when cells are grown under nicotinic acid, and you express the antibody that inhibits NIPX, you see that you inhibit growth. So that means that you are really in that rest. Okay, well, fortunately, the result of the reaction is color. So when you give the reaction, you get an intermediate that is color green, and therefore is very good. And well, it's nice, of course. They monitor visually whether the reaction is running well or not. So when you have cells growing in citrate, for instance, and then and then there's nothing, just a vector, we have the constant, we have the expression of this in your theory, then nothing happens here. But when you do the same and express the antibody inhibiting this enzyme, then you see a congregation following start there. As a control, this is what happened with Parkinson, right? So this situation is what happened with Parkinson, and this one is what happened with a conditional expression of the antibody that inhibits the same gene that otherwise it would well, the theory is not very good, but so another, say, added value of having this approach is that can make the variant the antibody that in this case, for instance, you can use to a GMP. And then if you make this combination between an antibody and GMP and you express that intracellularly against one of the proteins that are inside the cells, then what happens is that this enables you to see physically where the enzymes are inside the cell. And whether, for instance, they form clusters or they are distributed over the plate or over the tissue. So in this case in particular, what you observe is that if cells grow citrate, then you have a more or less homogeneous expression, this particular enzyme. But when you grow them in the proteinates, when you have the X, then the feature or the image becomes more granular. So it looks like if indeed you form kind of clusters of this enzyme that will make perhaps the reaction to run a bit better. Okay. So okay, so I want to recap a little bit what we have been discussing today. So first of all, when you need to construct a complex circuit or a complex device, then there are various things that you have to consider. Okay, so this will be like the ultimate thing that you generate. Well, first of all, you have to have very clear, a blueprint of exactly what you want to do, what is the relations between a block diagram, everything that is involved is particular situation. But then that's not enough. By the parts that are involved in the paper, remember the equivalence that we made the other day between components that are involved in electronic parts and so on. The same happens in biology. You have promoter, you have terminator, you have DMP, you have enzymes. You need a package of different things for assembling whatever say device again you. So, and what is very important is to have the right tools. It is not enough. A blueprint is not enough to have the parts. You have the right genetic tools to be able to put everything together. And one of the later talks in my discussion, it will be precisely about the genetic tools. Genetic tools are the essential part of the basis. The biology, in the sense that they are the ones that enable you to make all these type of combinations, all these type of groups to put different parts with each other, to make the different tests and so on. So this is basically the home lesson for today. That remember that for the sake of doing these type of things, you need to consider orthogonalization, you need to consider resource competition, you need to consider many of the things that we've been at today. And but that's not only that's not enough. From a theoretical point of view, at the end you have to get your hands dirty and assemble them with some source of the. Okay, so I think that will be most of the discussion for today. Let me just recap a little bit what we're seeing, what we're discussing. So remember that one of the basis of security biology is this concept of orthogonalization. Orthogonalization and organization. You will see that in later classes. There are other, say, key disciplines of this jargon, but organization is one that. Okay, organization, how to bring about organization, you can read like this at least, you can be by confinement of a reaction in a capsid, whether it's a viral capsid, with a oxygen, and you can think on recruiting or parts from promiscuous plasmids or promiscuous elements. You can think on extreme orthogonalization by thinking on orthogonal light, orthogonal specular or mirror light forms. But in more practical terms, orthogonalization is mostly about metabolic engineering, how to ensure that a module in a bigger metabolic engineering project is not or is associated or not to the rest of the metabolic network. And this is in many cases the instance when you have to think on how to isolate that part. And then for doing that, you can either try to isolate that part of the pathway with proteases, or isolate that part of the pathway with antibodies, or maybe you can figure out other ways to do it. One popular way to do it in the last few years is to use CRISPR-Cas, but as this is something that I haven't yet discussed, I don't want to make that. But you know, the CRISPR-Cas technology enables to produce precursors a la carte, because it can target different precursor proteins to different parts of the genome, and then you can also inhibit some parts of the whole metabolic network by taking specific complexes to target events. I will return to that in another class because there will be some time to discuss in detail the whole CRISPR-Cas saga. Okay, now what we have here now, I just want to that. Now, these are the assignments for next day. Okay. Let's see. Let's try to clarify this one in the different entities. Okay, so the other day we assembled 4 groups. So let me provide the information I have so far about the groups. Salud, Sigash. Sigash? Y tú lo entero también. Okay. We erase. He was frightened by synthetic biology or what? By the way, what what happens at the end of the course? Do you get a certificate or what type of value you get out of my lessons? But is that something that has some weight in your CV or in your? Okay, so you can here as a kind of hobby. Okay, that's wonderful. Great. Okay, so tool, then we have. Okay, so we have two positions here that perhaps.
So I'm not assigned to any.
They what?
Yesterday I didn't come for.
Oh, you didn't. Okay, so you are not in a group yet.
Yes.
Okay, so you have various. Maybe I should complete the names and then the ones who are unassigned can select a group to you. Okay.
Yes.
Okay, let's put that. Okay, then we have. Let's see. Number 2 is 17. Was 17. I'm sorry, my pronunciation may not be perfect. Yes. Wuhan. Okay, great. Varanasi. Varanasi. Odua. Ambash. Vaishnani. Okay, great. Vishal. No, Vishal. Okay, we'll describe the gender balance at some point. Okay, so number 3 and 4, I don't know, because you have not sent to me any feedback on that. Oh, so who were the other two groups? Okay, can you please write down there who are the members of the group? I will put that on writing in one of my sheets. Anita. So it's 3. Okay, very good. And you? Okay, very good, thank you. We have one other group. Who was there? So the other day we made 4 groups, right? So the other group has evaporated or what? So what's the other group? Okay, so let's go with the other. And don't be shy, I mean, you're not, I'm not going to retaliate you at all, just that I want to know how you can organize this properly. Okay, so 2 people who were not assigned to any group before, can you raise your hand? Okay, good, for instance, let's put it up. Okay, can you put your name here? Perfect. And then there is one, you. So well, I assume that I have all your emails, right? So excepting yours, I don't have your email. Okay, please do send me a message and give me your email so I can put you in the whole list of people. Okay.
I'm not assigned to that.
Okay, now we have a 4th group, so I think that the ones that are not assigned, please sign up here. Okay, please. So it will be 1, 2. Who else? 3? Any other? All others are assigned to our group already. Okay. Very good.
He wrote
Okay, so let's let's look at that. Okay, so is everyone assigned in this room? Okay, so let's try to maintain some issue, right? So I mean, it's not that you decide that this was first you are going to go, but for the sake of the dynamics of the course, let's try to keep more or less distinct the whole period. Okay, so I think that so it's the case that at the beginning, the first day, there's many people, and then after a few days, only the good ones remain. So I think that's something that will happen. I think that's okay, very good. So we have programs. I have prepared six or six papers, and then I will ask you to take your choice. Okay, so I started the other day. I don't know. We started this direction. Well, wherever we decided. Okay, so let's have take a look at the papers that I offer you for discussion. So if you remember, those of you that were not there before, I tell you for the first time. So the idea is that you take one paper, you read it, and then you discuss it in the group, and then you elect every day a different speaker that will present the paper question to the whole audience. And even though it's a bit intensive, for the week, every day we have the same thing. So tomorrow I have another 4 papers for each of you. The day after tomorrow, another 4 papers, so that at the end of the week, you all have the opportunity to have your moment of glory displaying the paper work. Okay, so I think it's important that a part of the way I tell you the classes, in some instances you go through the details because I can say, well, or peroxisomes are very important for this or that, but then it's good that you look at the details and then you decide by yourselves whether what I told you before is correct or has some basis or whether it's reasonable. And what to me is most important is that you not only make a summary what discuss or they tell you classes, but also to have a critical appraisal of whatever I tell you. So don't believe anything I'm saying. I mean, don't believe me about it. Be critical with everything I tell you, because as I told you the first time, the first day, the type of technologies and the type of ideas that we are going to discuss these days, you will be the most important beneficiaries of them. But therefore, you have the right to have your opinion. At the beginning, what I showed you about mirror life, and this is discussion about you have later. You say, well, this is complete bullshit. I'm not interested. I mean, this is something that perhaps has no basis. Or you may say, well, it's very cool. I think that's true. But as and the other day, and that at the end you have this critical appraisal and you read the paper, and you have a critical appraisal of what you think is convincing, what is not convincing, what could be improved, and all this. Okay, so what I propose you are these 6 papers. For some of them come from my own laboratory, others come from other places. So this one is I and my collaborators discuss what I told you at the end of today's election, namely how to make what we call phenotypic knockouts of selected metabolic pathways by targeting enzymes with camel-derived nanobodies. Have you ever heard about camel antibodies before? Because camel antibodies are a super useful tool, and you will see in subsequent classes that we have used them for many, well, we and others have been for many different applications. For instance, for microbiome engineering, one of the spin-offs, one of the applications of camel antibodies is to have them displayed on the surface of the cells. And then when you have a cell that is coated by one of these antibodies, then the whole antibody has the tendency to get attached to a surface that presents the corresponding antigen. And this is a phenomenal tool to make consortia and to stick bacteria. And it's really well, in any case. So just keep in mind that antibodies are very interesting tools synthetic biology for a large variety of purposes. Then, this one is really for only for the brain. Okay, so this paper is only for the brain. So in this paper, what we and others, and someone called Swayam Prabha, there is another name, is synthetic biology feed, and also some of the names of these papers you may see popping up the synthetic biology literature because they are names that over time have become quite important. So what we did at some point was to enter these cleavage sites of proteases in a super big protein complex that is formed by the growth EL, say protein complex. So the growth EL protein complex forms a chaperone system, and it's a system that many bacteria, well, basically all bacteria have to promote and to assist folding of proteins. So, well, I have the chance to that, but it's a complex paper, and what we can decide later to explain really go. Then this is not a paper, it's a review by Tom Ellis, that is one of the big names in synthetic biology, where he discusses some of the subjects, some of the ideas that I mentioned at the beginning of my lectures today about how synthetic implants interact or not with the rest of the networks, genetic and biochemical and regulatory networks of the cell. How can you handle the impact, introducing a new element, a new device, a pre-existing regulatory work. And well, this is a very interesting review. And the ones that wants to read this review, please do be nice and make a summary of the different strategies that are described in this review on how to handle this interplay between the basic metabolic, regulatory, etc. network and the new implants that we now fill the cells through synthetic biology. Okay, so we have three other statements here. So one of them is something that I discussed as part of the today is how to convert, how to make expression digital. So in expression, in most cases, occurs in a kind of continuous or recent fashion, and the promoter will have induction, and then over time, how to make something digital that you have no expression and what, or basically you go from 0 to all. So this is a paper that we had a few years ago, and I think it's quite interesting from the point of view of how to engineer a device that makes assistance close to what you see in the cells. Then this other one that is an earlier paper that goes together with the paper on the represilator that we discussed the first day, and so the paper on the protomedicines. So this paper comes also from year 2000, and it's the third paper that is considered foundational for synthetic biology. So in this case, what it is the engineering of a biological system where you enter a degree of feedback control. So feedback control, I don't really remember the first day I showed this picture with the how engineers figure out ways of controlling the flux, a liquid in pipe, and having supposed rotating and so well. This is something that is one of the examples that the same concept can be implemented in a biological system. This is the work of Serrano and Bexel. This is again one of the semi-obligatory readings for those who are interested. So the origins of the readings. And finally, and this is like more visual, here is a review on how to exploit viral capsids for designing nanocompartments that are designing enzyme confined compartments that act as catalysts. So that's something that. Okay, so we have 6 papers and we have 4 each year. All these papers are available in a file that is in the Dropbox, say, the address that I share with you. So we have to upload it for the yes. But if I have 6, I will have 4 for. So I don't know how I got it. We make a kind of say lottery, or we record, I say you take this one, you take this one, whatever. Lottery. Okay. Okay, good. Let's see. Okay. Or we self-paper, right? So let's do this thing about lottery proper. Okay. Actually, okay. Don't open it yet. The sense. Okay. Okay, then it's here. And then Shingu. Okay. Okay. This shampoo. No. Okay, great. So let's see what it was. Okay. Yours. A4. Okay, then please get this market. 2. You get this one. I'm sure this is the most effective. And you are very good. Okay. It's 6. It one. That's it. Okay. This one. This is also complicated, right? So, and so this is that's correct. Okay, great. So the paper that I have handled. So you have here the 4 papers, right? So please, as I said, get together, discuss about this paper, and design one, say, liaison or presentation of the paper. Obviously, it has to be different every time. Although with this spread, so first, I think, speak to us. And so little by little, all of you have the chance to exercise. Don't be shy. You know, we are among friends. So because that you already might be interested to start as the discussion. We need more discussion. Ladies and gentlemen, I get very bored to keep on talking and talking and talking and talking. I have heard myself many times, so I'm very bored. So I think that we should start maybe after the coffee some kind of discussion on different some of the things that we have heard today, and start getting some feedback on your opinion on how far synthetic biology can go, how far it can influence the way we practice biology and statistics. And I see your opinions are very important. If you are coming to the field, I mean, please think on what would be your reaction to this idea that you can engineer biogas systems, because we are biogas systems. And you is not complete, it's not alien, it's not like in a radio. I mean, building a radio has no influence in your daily life, but you know, telling you that you can engineer biogas systems will have an influence in your life. It is important that you think not only as a student, an observer, but also as an active participant. Okay, so I think that the coffee is there, more or less, and we break until 4:30 or so, and then we will be first from here, whatever